Here we go with sensory mechanisms 8.10. Let's start with the ear, and we're talking about the sensation of hearing. So this sensation is accomplished by these mechanoreceptors that are located in the inner ear. And basically, when we're talking about sound, it's basically the hairs bending. Um, and so let's start off by taking a look at the actual structure of the ear. So the outer ear, its function, that part is for the collection of the sound waves. So those get trapped in the ear. You can see by the shape of it. It cups it. It funnels those sound waves in and brings them inside. So this outside part is called the pinna. It's made out of cartilage. Continues to grow throughout someone's life. That's why people can have very large ears. But it acts as an antenna and collects those sound waves. And then we move into the auditory canal, and this is concentrating these, concentrates the energy as it moves toward this middle ear. So the outer ear is made up of the pinna and the auditory canal, and then you have your middle ear right here, and you also, in this outer ear, you also have this tympanic membrane, and it's also called what we call the eardrum, and that converts the sound waves into vibrations, and then so we move into the middle ear, and what this one does is to amplify the energy that's traveling toward this inner ear, and the middle ear. So when we look at so here's our tympanic membrane here. And when you hear someone talk about that their eardrum ruptured, it happens a lot with ear infections. That's what's breaking right there. That's what's perforating, is that tympanic membrane. And so the middle ear is made up of what we have the hammer, anvil, and stirrup, these structures. But you have the malleus, the incus here, and then the stapes. Okay, so this structure right here makes up the middle ear, and they convert that sound wave into vibrations. And then we have this eustachian tube, and that connects with the throat, and it acts as a pressure valve for the ears. And one of the problems with babies is this eustachian tube hasn't dropped, and so it's more flat and level, and they're drinking their milk, and bacteria builds up in there very easy and then presses here against that tympanic membrane as this swells. But it acts as a pressure valve. And then we have the oval window here, and that actually converts the energy into a fluid wave energy. So then we move into the inner ear, and this is where that transduction of the fluid wave, so the fluid waves occurred there in the middle ear, were converted to that, and now this converts into the electrical energy that's going to go off to the brain, so that the brain can actually interpret what's going on. And so this cochlea, you'll hear people talk about, it's a lot more common now for people to have cochlear implants, and that's when you see they have like a connection on the outside of their head, and that's what they're doing, this connection here with the cochlea. So this cochlea is a snail shell shape, and so it's filled with perilymph, and it's a fluid, and it's used to make these ripples, and it's made up of the vestibular canal right here, and then the tympanic canal, and the cochlear duct. So you can see all these, there's a whole lot to. Here's that oval window, here's that stapes, so here's your middle ear, and then coming into the inner ear, you've got all of these, the tympanic canal, the cochlear duct, and then here's where the perilymph, this is where the liquid is formed. And the basilar membrane contains, that's where the actual hairs are. And the hairs bend, and when the hairs bend, they cause the neurons of the auditory nerve to create an action potential, to create electrical energy. And this round window, the purpose there is it absorbs that ripple. So you can see it comes in here, and this is where that's moving along here. So this round window absorbs the ripple so it doesn't create waves in the opposite direction. All right, now when and then so here you can see these hair cells in here, and just so as the hairs bend, they create that electrical impulse. So let's look at volume. We also call volume loudness, and what we're really referring to is the height of the sound wave. So the larger, the blue is loud, so the higher the sound wave, the louder the sound is, and the green is the soft wave, is the softer sound, sorry. All right, and then pitch is referring to frequency. So this is the number of sound waves that pass a given point in a certain amount of time. So in terms of hearing, this refers to the number of sound waves that hit the tympanic membrane per second. It's measured in hertz. Human hearing is between 20 and 20,000. You don't need to know that, like memorize it or anything. Most animals can go much higher than humans. That's why you can a sound can be created, a dog whistle can be created, and we don't hear it as humans, but dogs certainly hear it. And you think about hearing, humans have lost some of their hearing because we don't need to hear as well, but a blind person has tremendous hearing because they need to, because they don't have their other senses to pick it up. Balance and motion, these are all related to with the hearing. If someone has a hearing an ear infection, everything's thrown off. And so again, this balance and motion, this is related to those hairs bending in the inner ear. You have this vestibule, and that's where the lymph, the paralymph reservoirs are. Whoops. And then these semicircular canals, there's 3 on each side of the head. They're actual organs. They detect sensations. They're filled with this fluid. And so when your fluid is off, or the pressure is off, it sends all of that, your balance off. The movement of the body causes this paralymph to flow through the canals and bend these hairs in there. And so and it's the bending of those hairs that cause the depolarization in the neurons, and this energy of motion is converted to electrical energy. So remember, we have to have the electrical energy that's going to end up ultimately going to the brain. All right, now with taste, we have these chemoreceptors that are actually on the tongue, and it's accomplished using receptors that are in the nose. So these are for olfactory, means smell, and gustatory means taste. So we have these taste buds, and chemicals are detected by different neurons upon contact. We have 5 taste senses: sweet, sour, bitter, salty, and then savory. And believe it or not, taste is 80% smell and 20% taste. That's why when you have a cold and you can't smell anything, food seems to be tasteless. And then here's just those chemoreceptors along the nasal cavity. All right, now let's look at sight. So the sensation of sight, we have different types of light that light detecting structures. The ocelli are seen in cnidarians and bivalves, and they have this there can be an eye cup. Planarian have these eye cup here. Platyhelminthes, platy of course meaning flat, helminth meaning worm. So these are the flatworms. Planarians are examples of that. And they have these photoreceptors. And then we can have, there can be compound eyes, which you find in invertebrates like insects, or you can have, like you can see, these very complex, lots of them all working together, or you can have the single eye like you have with mollusks and vertebrates, and we can see definition with that. looking at the actual structure of the eye, we have the sclera, which is the white of the eye. The choroid contains the blood vessels and the black pigment for reducing sun glare. The conjunctiva, that is involved with mucus production to keep the eye moist. And so when someone has conjunctivitis, or we call it pink eye, that's the inflammation of that conjunctiva. The cornea prevents debris from entering the eye. And then we have the iris here, that's the colored portion, and then we have the retina back here on the inside, and this is for photoreception, and we can have rods versus, we look at rods versus cones. The rods are for seeing black, white, and shades of gray. The cones are for seeing color. And so we can tell if something can see in color because we're looking if they have rods and cones. And so we can tell what that organism can actually see. Then there's also, if we go back here, we also have the lens. And that lens is what accommodates, what focuses the light. And then the different problems that could be, you could have stigmatism, which is a misshaped lens, myopia, where you can't see things, where you're nearsighted, hyperopia, farsighted, and so if your lens gets clouded, that's where people have cataracts. Or if there's too much vitreous humor, which is the liquid that basically gives the eye its shape, if there's too much of that, then you can have a condition called glaucoma, and it creates too much pressure in the actual eye. The optic nerve, there's one for each eye, so you can see these nerves. So here's your eye, and that nerve comes out and connects there to the brain, sends the signals to the brain. There's one for each one. And I think that's, we do have a little bit more left here for locomotion, but I think we'll throw that in with part 11, and we'll do those two together.